Welcome to the CannMed Coffee Talk podcast, where we talk with the leading minds in cannabis science, medicine, cultivation, and safety testing. I am your host, Ben Amirault. I'm the marketing manager at Medicinal Genomics and proud member of the team that puts on the CannMed conference every year. Okay, big announcement to make at the top of this episode. The dates for CannMed 2021 have changed. This year's event will take place September 29th through October 1st at the Pasadena Convention Center in Pasadena, California. The CannMed Medical Practicum will be held on Wednesday, followed by the full conference on Thursday and Friday. This decision was made after careful evaluation and consideration by our management team and various state officials, and it was done in the interest of safety so that we can hold a safe, in-person CannMed event. All sponsors and speakers announced thus far have been notified of this change, and we will continually update CannMedEvents.com should conditions change. Any current ticket holders who are not able to make the new dates can receive a 100% ticket refund. Go to CannMedEvents.com/COVID-19 for details. With that out of the way, let's get to today's guest, Professor Raphael Mechoulam. Professor Mechoulam is the first researcher to isolate THC and CBD. For that achievement and the decades of research into the endocannabinoid system that followed, Professor Mechoulam is often called the Godfather of cannabis research. Since his research began in the early 60s, Professor Mechoulam has received numerous academic awards. Most recently, the Harvey Prize from the Technion, Israel Institute of Technology, in the human health category. It's a pretty big deal when you consider more than 30% of Harvey laureates since 1986 have gone on to receive the Nobel Prize. Professor Mechoulam has presented at every CannMed event, either in person or via submitted video, and he is the chair of our scientific advisory board. In our conversation, we discuss Professor Mechoulam's incredible career, the disappointing lack of cannabis medicine clinical trials, the surprising fact that the two major endocannabinoids, Anandamide and 2-AG, have never been administered to humans, the challenges of researching the entourage effect, the benefits of cannabinoid acids, the newly discovered anandamide-like compounds that could be used to treat addiction, and much more. Before we get to that conversation, I would like to thank this episode's sponsor, Cannabis Science and Technology. Cannabis Science and Technology focuses on educating the legal cannabis industry about the science and technology of analytical testing, quality control and assurance, extraction, cultivation, and processing and manufacturing. In October, Cannabis Science and Technology launched a new brand, Cannabis Patient Care, which is an educational resource for patients, doctors, nurses, pharmacists, and caregivers on the latest research, benefits, and treatment options cannabis provides for a variety of illnesses. Go to CannabisScienceTech.com to learn more. And lastly, it wouldn't be the CannMed Coffee Talk podcast without some good coffee. And for that, we always turn to the Hemp and Coffee Exchange. If you don't know, hemp coffee is a healthy, delicious, natural product rich in trace minerals and nutrients, providing sustained energy without the crash of regular coffee. For more information, check out hempcoffeeexchange.com and use the promo code DRINKHEMP to get 10% off your purchase. Okay, and without any further ado, please enjoy my conversation with Professor Raphael Mechoulam. Good evening, Professor Mechoulam. Thanks so much for joining us.
It's a pleasure.
First, I want to congratulate you on the Harvey Prize that you recently won for your life's work on the endocannabinoid system. Very well deserved, and dare I say, long overdue. You've mentioned in the past that there was really not a lot of interest in exploring cannabis when you first got started in the 60s. But what about cannabis inspired you to start investigating it?
Well, you see, the chemistry and pharmacology of morphine, cocaine. They are the major addictive drugs. These were known starting from the 19th century, not only the chemistry, but extensive pharmacology, extensive knowledge. This was not the case of cannabinoids. And the reason was that, well, there is a chemical reason. It's really a big mixture, very difficult to isolate the compounds. And two chemists, major figures in the chemical field, had worked in the 30s, early 40s. And because of the war, they stopped. And later on, it was not taken up, probably also because of legal reasons. It's very difficult for an academic lab to work on compounds that are legally restricted. One probably has to have somebody looking around to say that students don't take it or whatever. Anyway, there was one group, not a major group in Germany that was working, and maybe a few others, but essentially it was a field that nobody was working then. Although, as I said, morphine and cocaine, chemistry, pharmacology, clinical trials were well known decades previously. So I thought it's a good idea to go ahead. And I was lucky that I could get a hold of cannabis. I assumed that I wouldn't have been able to do that in the US because of legal reasons. And here I just went to the police after a phone call from the administrative head of my my institute. They asked, can they depend on me? And he, without even knowing me, said, of course you can depend on Mechoulam. So I went there. They gave me 5 kilos of hashish, and we started work. I very much doubt whether I would have been able to do that in most other places.
Sure. And so you've mentioned that there was a lot of work going into other drugs like morphine or heroin. How come there was no concern studying those drugs, but there was with cannabis?
Well, I was not interested in those. You see, I didn't have a group doing research. I had to do that with my own hands, and a friend of mine, with the two of us, Yechiel Gaoni, were close friends, and this was it. So we couldn't compete with major groups working in anything else. I thought that the best idea is to work on topics that we can do without having a major group, without having a lot of money to do it.
So now you started studying in the 1960s, and when you did that, did you think that you would still be working on cannabis today, or that we would be where we are today?
No, not really. I thought that we'll do the chemistry for a couple of years, and that was it. Then it turned out that we always found new topics of quite a lot of interest. So we had to move along all the time. And so the beginning in the 60s, early 70s, worked mostly in the chemistry, and then on the pharmacology and metabolism. Then a little bit later, or quite a bit later, it was realized that there are receptors. So we went ahead and looked for compounds that are formed in the animal body, animal brain, if you wish. And we found that, yes, there are specific compounds that add, and here we had a new physiological system, a completely new physiological system. Nobody had done anything on it because it was not known. And it turned out that it is of major interest. As a matter of fact, people at NIH wrote a review at that time, I remember, and wrote that the endocannabinoid system is involved in almost all human diseases. Now that's a very, very, it's a thing that one cannot really accept so easily. Even the major neurotransmitters like dopamine and serotonin, one cannot say that they are involved in essentially all human diseases. Now, this being the case, there is a lot of work. So we went ahead doing a lot of additional chemistry, additional pharmacology, to a large extent, clinical trials that we could do. And so it developed and expanded, and that was it.
Now, you say it's sort of hard to imagine that the endocannabinoid system is involved in every disease or every body system. And it is quite amazing. To what extent is it involved, though? Is it essential, or is it sort of just a contributor?
Well, it seems we don't know about all diseases, certainly, but it is definitely involved in many, many of them. And so I can't really define whether in a certain disease is the central one or not. We have to go and look at each one of them. And but it is. Now, we also have a problem, a major problem, and it is that and the two endogenous cannabinoids that we discovered, Anandamide and 2-AG, they have never been administered to a human. So what happens is that we can only say, well, it works in mice in a model of a human disease and so on. So we have a problem there. But that's where we stand. But even so, there is a lot of work. Our paper, in which we described the isolation identification of anandamide, has been cited more than 6,000 times, which is very unusual for a paper. And the same is true for 2-AG. There are 2,000-3,000 times being cited. So there is a lot of interest in whatever these compounds do or whatever they are involved into.
So that shocked me when I learned that anandamide and 2-AG have never actually been administered to humans. What is the reason for that, especially when there's plenty of examples of cannabinoids, which are, created by the plant to sort of mimic these compounds, have been administered? Why not the real thing?
Well, I can only imagine. See, in order for a compound to be administered to a human, one has to do a lot of preliminary things, a lot of toxicology, of a lot of other things. And for example, a drug, in order to be administered to a human, one has to spend a lot of money. And this is done usually by companies. Well, there is no company involved because there are no patents on these compounds. And if we had patents, they would, we published these things in the early 90s, so that's far away. And so no pharmaceutical company is interested. And the governments do not do that. So here we have a strange situation, which a compound, which is not toxic, or two compounds, which are not toxic, which are very much involved in our physiology, which have a lot of effects that are very relevant to what we're doing. These compounds have never been tested in humans. Strange, but that's it. Now, I can't do it because we don't have the amount of money needed for that. We don't do toxicology, and these things cost millions. So there we are. Strange situation, but there we are.
Wow. That almost seems criminal. I mean,
Yeah.
It seems like a tremendous opportunity missed, and that someone should really take up that mantle.
Well, what companies are doing, they're trying to work on the enzymes that synthesize anandamide and 2-AG. They can block, and so the body doesn't make anandamide. Or on the contrary, they block the enzymes that metabolize anandamide and 2-AG. So the levels of anandamide and 2-AG go up. So here we have something that is done, not straightforward, a little bit in a strange way. Or, for example, runner, if somebody runs and gets exhausted by running, so one would expect him to be, well, kind of, not very happy. No, we know that there is a runner's happiness or something like that. And this is due to the enhancement of anandamide. If a person does a lot of physical exercise, the anandamide goes up and the person is in a better mood. So this is not a direct way of increasing anandamide. Or if one gives cannabidiol, a group of found that if one administers very high doses of cannabidiol, in this case to schizophrenic patients, the cannabidiol acts on the schizophrenia. It's a pretty good anti-schizophrenic compound, but its effect is also an anandamide. Anandamide goes up quite a bit. Maybe that's the mechanism, the effect, but it goes up. So this is one way of changing the levels of anandamide in the body.
That's great. And as a runner myself, I can say that I get healthy doses of anandamide after every run, which keeps me coming back for more.
You smile after that.
Of course, yeah, I love it.
Good.
So I was hoping we could maybe take a step back for maybe people who aren't so familiar with the endocannabinoid system. Could you give sort of a brief explanation of, what are anandamide and 2-AG doing in the body? And why is it beneficial to have good levels of it?
Well, we don't have a full picture of what they do, but most definitely they are the major compounds in the body that stimulate the endocannabinoid system. And the endocannabinoid system is involved in a huge number of disease states. So the body produces anandamide when one has pain, for example. Without anandamide, the pain will stay for a longer period of time. So you get pain, and then the body starts producing anandamide around the painful place of the body, and the pain goes down. Or in the brain, it acts on the pain effect in the brain. After all, if we have pain somewhere on the body, the effect is through the central nervous system. So anandamide acts on pain. As a matter of fact, this is one of the major ways we lower pain. But anandamide is not, does not stay around for a very long time. It is produced when and where needed. It acts, and it is more or less broken down immediately. So the same is true for 2-AG. So here we have a compound which is a reaction, if you wish, to physiological states. And this is the same is for many other disease states.
So it's really not produced unless there is some sort of stress or some sort of stimulus.
Well, not a stress, obviously, but also it's part of our individuality, if you wish. Some people there is a difference in personality. And one of the reasons for the difference in personality is the endocannabinoids. A person with, let's say, a happier personality may have more anandamide than a person that is not. So it has to do with what we are.
Excellent. And now, so where does cannabis play into this? What are the compounds in cannabis that sort of mimic the effects of anandamide and 2-AG?
Well, I don't believe that the plant really wanted to do anything to us. It produces cannabinoids because of its own needs. You see, we are not different from most animals in our physiology. Our physiology contains a lot of compounds, hormones, neurotransmitters, and so on, but also, so does a horse, a pig, whatever. This is not true for plants. plants don't have the same chemical thing as other plants. Every family of plants, or almost every family of plants, produces different compounds that act for them. For example, a certain plant is attacked by some insects. Over the years, over the ages, the plant has started producing compounds against their specific enemies. And so every plant family has its own group of compounds, which in many cases are specific for that plant or for that plant family. And so is with the cannabis group of plants, there are 2 or 3, they make compounds of this type. No other plant does it, only plants that belong to this particular family of plants. So they are produced for their own reason. We're not exactly sure why they produce them, whether it has to do with something to do with the soil they were originally grown on, or it's something that has to do with fungi or microbes. We have seen that some of these compounds have antimicrobial effects and so on. So we don't know exactly why cannabis makes the compounds, but it does. And these compounds are specific, and there are about 100 of them. 2 or 3 are the major ones, and there we stand.
Excellent. And in preparation for this conversation, I went back and watched your CannMed 2019 presentation, which I will put in the show notes for everyone else to enjoy as well. And at one point, you were very, very vocal about the need for more clinical trials in terms of whether it's cannabis medicine or cannabis-based medicine or even the endocannabinoid system. I was wondering if you could speak to some of the clinical trials that have happened, and if there were to be more future ones, where should researchers sort of focus their efforts, in your opinion?
Well, quite frankly, it's a shame. It's really a shame that we don't have more clinical trials. A huge number of people take cannabinoids or take a plant extract for a variety of diseases. In most cases, without clinical trials. We know that cannabinoids act on pain, but pain is not a single symptom of a disease. There is pain from the different parts in the body and so on. We don't have large clinical trials. A lot of people take cannabis for cancer. Well, first of all, cancer is not a single disease. There are many types of cancers. And so there aren't any real major clinical trials with cannabinoids. And so people are taking cannabis for cancer without really any background, real background. It's a shame. The THC or CBD act better on one cancer rather than another one. Glioma, cancer in the brain, is not the same as a cancer of any other organ. They're different types. And we don't know. Just people are taking them because they've heard that that helps. We're the 21st century. It should not be that way. And the reason is that clinical trials today, in the Western world at least, are undertaken by companies. Governments are not doing clinical trials. But in this case, companies are not involved because there are no patents, and the company does not want to spend many millions of dollars or pounds, whatever, just to try something in for a publication. So here we have a situation which is unacceptable. I believe that the governments, through their ministries of health or whatever, should do clinical trials because nobody else is doing them. And NIH normally does not do clinical trials. in this case, I believe that NIH should do clinical trials in order to know where we stand. A lot of people say that cannabis can cure cancer. Well, I'm not sure. It acts on cancer. It blocks in animals cancer development. So it has been shown that cannabinoids can prolong the life of people that have a certain type of cancer. There is no proof that it's a anti-cancer drug. I believe that it helps along, but it's not anti-cancer drug. But all these things have to be investigated in human patients and not in animals. There we are.
Wow. So it seems like a common theme here. There's just not the investment from companies to look into it because there's not a lot of opportunity for them to make money on it. So I think what we need is some philanthropic person to just donate a whole pile of money to investigate these things.
Yes, all governments. I mean,
Sure.
after all, millions are using cannabis. The government should probably look into a problem that's a wide problem, not a few individuals. We're speaking of millions.
Yeah. Well, I think we need to get them on board with decriminalizing it and descheduling it first. So I guess baby steps. And I imagine another thing that's sort of complicating the ability to do real good research into the effects of cannabis medicine is this whole idea of the entourage effect. So I wonder if you could speak a bit to that.
Well, most cannabis users say that in total extract of cannabis is much better for them individually than the specific cannabinoid, CBD or THC or whatever. And it seems that this is true. And we have done a few investigations in this along this line. And yes, we have seen that apparently the extract contains compounds that affect the THC or CBD actions. And I'm, again, sorry to say that although this is widely accepted and the term entourage effect, which we coined 20 years ago, is widely used, but there are, again, no trials with, even in animals. We don't know whether all the terpenes that are present in cannabis together as a group have to do with the entourage effect, or only 2 or 3 have specific effects. We don't know, and it's a pity because we could have much better cannabis if we had the correct knowledge.
And I got to imagine a compounding complication there is just the variability in different cannabis strains, or even, plant to plant of the same strain could have, different concentrations of different compounds just based on how that plant is grown. So I would imagine in order to do some real research into what combination of cannabinoids is contributing to this effect, you need sort of standardized formulations to look into something like that.
Most definitely. We, for example, one can imagine taking THC with 2 or 3 or 4 well-defined terpenes or flavonoids is better than THC itself, maybe. But all this has to be investigated. And it has not been investigated. The number of terpenes is quite large. We know the structures, we know what terpenes are present, although they differ, of course, from cannabis sativa and cannabis indica and so on. And indeed, in Israel at least, cannabis patients or people that take cannabis usually take cannabis indica in the evening and cannabis sativa in the morning because of the different effects. But we don't have enough data to say why.
So we're getting close to the end here. I did want to give you an opportunity to speak about what research you're doing now.
Well, we are working now mostly on anandamide-like compounds. And we've published that our body, in addition to anandamide, makes a lot of compounds which are chemically closely related to anandamide. Anandamide is a fatty acid bound to a derivative amino acid. And the body has quite a few fatty acids and quite a few amino acids. And so it makes a lot of these compounds. We have about, I don't know, something like 100 compounds of this type. And they're not made by the body because it has nothing else to do. They're made for a specific purpose. So we found, and is always a collaboration with pharmacologists and clinicians, found that one of these compounds is very helpful in brain damage. Another compound we found is very efficient in osteoporosis. And over the last few years, again, a few groups of friends, a group in the US, a group in Canada, a group in Italy, and our group, together we are working on addiction. The mechanism of addiction is not actually well known. For many years, it was believed that addiction is the psychological effect only, and that's it. Well, no, over the last 15 years, 20 years, it has been recognized that addiction is actually a kind of a disease, and there are all kinds of changes that happen in the body, and we don't know much about it. So we started looking together for the changes that happen, and we were quite surprised to find a compound that our body makes and tries to block nicotine addiction. We started working on nicotine addiction and have moved to opiate addiction. And we found that there is a compound, again, anandamide-like compound. It's a compound made from oleic acid and glycine, two very well-known compounds. So it's the amide of oleic acid with glycine, and OG, oleal glycine, blocks nicotine addiction. It doesn't always succeed, but it blocks nicotine addiction. The body makes it. What's interesting, probably even more important, that this particular compound we found blocks the withdrawal effects of both nicotine addiction and, much more important, of opiate addiction. And this was quite a surprise. Many addicts are afraid to stop using opiates because of the withdrawal. They are very afraid of the withdrawal, which is really a very painful state. So here we have a compound that our body makes, not enough, but it makes, and in animals at least, blocks the withdrawal of nicotine, the withdrawal effects of both nicotine and opiates. So I hope that this will be investigated by a company. The company, as a matter of fact, has been funding a part of our efforts in this field of all four groups. And I hope that we can contribute something quite practical, which is far away from the cannabis in the plant, but it's still closely related to anandamide. So I hope that this is something that will happen. We also work on derivatives on cannabidiol. Cannabidiol, we didn't speak about it too much. It's an interesting compound. It has a lot of effects, all of them positive, almost all of them positive, and it works in a lot of disease states. And we thought that maybe we can make a more potent cannabidiol. Cannabidiol in schizophrenia, as I mentioned to you, has to be administered very high doses, about 800 milligrams a day, acts on schizophrenia. At least that was published by a group in Germany. So we thought that maybe we can make more potent cannabidiols. And yes, we have synthesized quite a lot of, and published quite a lot of cannabidiol derivatives, and some of them are indeed better than cannabidiol itself, and the company or two companies are working on cannabidiol derivatives.
Wow. If we could go back just for a minute to talk about those anandamide-like compounds, is it possible that there'll be other compounds in cannabis that are closely related to that, much like happens with anandamide and THC?
Well, anandamide and THC are almost equivalent in their activity, although it's like completely different chemically, but their activity is closely related. The major difference is that anandamide is broken down almost immediately, and so we don't see in animals the same effect that we see with THC. But the effect, the basic effect, is essentially the same. Do we have compounds, let's say CBD, acting like an endocannabinoid? Well, they are different, but maybe cannabidiol is mimicking, maybe it mimics one of the endocannabinoids which have not been well investigated. I said only anandamide and 2-AG have been investigated, quite a few additional compounds. So maybe here we have something that has to be discovered.
All right, still lots of work to be done, I'm sure. And another thing that jumped out to me when you were talking was this need for collaboration and working with all these different types of groups, which is very near and dear to our hearts here at CannMed. And it's what we try to do every year, is bring together everyone who's working on this plant or working on this problem and bring them together so we can push everything forward. So it's good to see that we're on the same page with that.
Well, in my view, it's essential. I like working with friends, with colleagues, and it's essential. All the problems that come up with anandamide, with cannabinoids, their chemistry, their pharmacological effect, their clinical effects, and so on. There isn't a single group that can do all these things simultaneously. So the best thing is to do it with colleagues and friends. And we, well, when there were meetings, not anymore personal meetings because of the corona, but we used to sit together, drink a lot of coffee, shout at each other. Well, not throw stones at each other, but shout at each other. And at the end, come up with a suggestion together how to do that. And the addiction thing is probably typical. The compound was the compound or that acts on addiction was identified by a group in Italy, my good friend Di Maso. It was investigated in quite a lot of details in Canada. A group in the US is also doing work on it, and we've been doing the chemistry. So I don't believe that a single group can do that.
Absolutely. With a problem this big, it's going to take all of us. So I know we're
the problem is to have good friends and colleagues.
Absolutely, and hopefully we can create a venue to create new friends and colleagues at CannMed. So, Professor Mechoulam, I want to thank you again for taking the time to speak with us today. It's been a pleasure, and thank you for everything you've done to help CannMed over the years.
In your questions, first of all, thank you very much. In your questions, you asked about the cannabinoid acids.
Yeah, absolutely. If you have the time, I'd love to learn more about that.
Well, you see, the plant doesn't produce THC or CBD. The plant produces their precursors, which are cannabinoid acids, which are not stable. As soon as they are produced, they slowly break down. And there is a cannabidiolic acid, which forms cannabidiol, THC acid, which forms THC. So these acids have not been investigated thoroughly because they're not stable. And a researcher cannot really work with unstable compounds because he doesn't know whether the effects are due to the original compound or the compound that has been formed. But so we managed to stabilize cannabidiolic acid, and we work with a stable cannabidiolic acid, and it turned out that it is of a considerable importance. It has a lot of effects, and companies looking at these effects and so on. It has to do, again, in collaboration. We found all these things. It has to do with anxiety. It has to do with some other effects of this type, but it also has to do with pain and inflammation. And so there is a lot of work going on now in my lab together with friends. And I believe that these compounds may turn out to be as important as CBD and THC, hopefully.
Now, is it that they are more effective at doing the same thing that the non-acid forms do, or is it a just completely different effect?
No, they seem to be more or less as CBD. For example, CBD acid, the stable CBD acid, has turned out to be a very powerful in pain, more powerful than CBD by far, more powerful than CBD. And a colleague in Canada published that, and he found that it's many times more powerful than CBD. But surprisingly, he found that it acts better or essentially only in male mice, but not in female mice, and I don't know why.
Wow. Well, that's certainly interesting. So it could be that we see more CBDA products on the market in the coming years?
Well, I hope so. A company is working on those compounds, and they support some of our work, and they're trying to develop these compounds, at least maybe some derivatives of these compounds. And I have not seen any toxic effects. So I believe that these compounds may reach the public as official drugs in a few years. I hope so.
Excellent. All right. Well, before I let you go, is there anything else you'd like to discuss?
No, as you mentioned at the beginning, we thought that it's a small project, and it has turned out to be a major project. And when we thought it worked, there was nobody working. Now there are probably hundreds of researchers, people that develop them, people that sell these compounds. And so one never knows exactly what will happen when one starts a new project. But when students come up to me and say, what should I choose to work on, a new faculty member, I always say, start with a problem that nobody else is working on. You have a better chance of succeeding than being just one of a major group of researchers that are doing work on something that's reasonably well known.
Certainly good advice. All right. Well, thank you again, Professor Mechoulam, for taking the time.
You're most welcome.
We hope to see you soon.
Bye-bye.
I hope you enjoyed my conversation with Professor Raphael Mechoulam. Check out the links in the show description to learn more about the topics we discussed. And thanks again to our sponsor, Cannabis Science and Technology. Our next episode will drop February 10th. In the meantime, please go to canmedevents.com/coffeetalk and sign up for email updates. That will enter you into a drawing to win 2 tickets to our CannMed 2021 VIP dinner and keep you up to date with all things CannMed 2021. If social media is your thing, you can find us on Facebook, Instagram, Twitter, and LinkedIn. Just search for CannMed Events. And lastly, if you are listening via a podcast app, go ahead and hit the subscribe button so that new episodes automatically download to your device. And please leave us a 5-star review as well. All right, that's it from us. Stay safe, stay healthy, and be sure to come back for the next episode of CannMed Coffee Talk.